Mr. V's Biology.
Science rules.
Mr. V's Biology.
All right, so we are in this Amoeba Sisters video recap of meiosis. The purpose of meiosis is to make gametes, also known as sperm and egg cells. In humans, your body cells have 46 chromosomes. How many chromosomes are in sperm or egg cell if when they come together to form a fertilized zygote, there are 46 chromosomes? Write the correct number of chromosomes next to the sperm and egg. So I have 23 or 46. And so I know that in every single one of my body cells, there are 46 chromosomes. That means that in sperm, there is 23. And in egg, there's 23. That makes sense. When sperm and egg come together, that makes 46. And those start duplicating, which makes us. Number 2, interphase must occur once before meiosis can happen. What would happen if interphase didn't occur first? If it did not occur first, the DNA and chromosomes would not copy to go into the new cells. Remember, interphase is where DNA copies itself. And so that's where those chromosomes are copied before the new cells are made. So if interphase doesn't happen, then there's no DNA or blueprint to go to the new cells. Number 3, a cell that begins meiosis has 23 chromosomes inherited from the mother and 23 from the father. In the process of meiosis, chromosomes begin to match up in homologous pairs. How would you know if 2 chromosomes were homologous? So in this unit, we're realizing that there's lots of vocabulary. And so that's actually the next couple lessons is all about the vocabulary so that we can learn it. So homologous, homo means same, logos means pair. So homologous means same pair. So how would I know that these 2 chromosomes were in the same pair? They would be the same size. They're the same shape. And they have the same genes. Genes is a chunk of DNA that codes for a trait. Okay. Number 4, crossing over is very important. It's a very important event in prophase 1 of meiosis. What happens during crossing over and what is the significance? The chromosomes from the egg and sperm swap DNA, making unique chromosomes. Number 5, meiosis does PMAT twice. That's prophase, metaphase, anaphase, telophase. That means there is prophase 1, prophase 2, metaphase 1, metaphase 2, and so on. If the cartoon below has chromosomes in the middle, how would I know whether it was in metaphase 1 or metaphase 2? But it's in metaphase 1 because there are 2 pairs. Metaphase 2 would have 1 pair. And so they also taught about non-disjunction. So what would occur if non-disjunction and the affecting results? So non-disjunction, the chromosomes do not separate correctly, making the new cells not have the correct amount of chromosomes. Meiosis makes unique cells, and mitosis makes identical cells. They both involve DNA and chromosomes. Mitosis is made through asexual reproduction, while meiosis is made with sexual reproduction. The DNA replicates in interphase. That happens before both mitosis and meiosis. Mitosis is one stage, meiosis is two. Creates two daughter cells would be mitosis. There are 4 daughter cells in meiosis. They both start with one cell. Mitosis has the same number of chromosomes while meiosis has half. And then we have these two vocab terms, which again we'll go over in more depth tomorrow and Monday to help us review all this vocab. I remember as diploid double, haploid half. So mitosis makes diploid cells, where meiosis makes half the number of chromosomes in each cell. So those are called haploid. Okay, so I will pause it on this screen and let you make yours look like mine. Okay, so number 1, view the cartoon below. Describe how this cartoon illustrates interphase. Well, interphase is where the chromosomes are replicated or copied. So that's why we have this chromosome on the copy machine. How many times does interphase occur before mitosis? That's 1 time. Meiosis, 1 time. So number 2 wants us to, or number 4, draw a chromosome before interphase. Label the chromatid and the centromere. So here's my chromosome. The centromere is in the center. Centromere in the center. And then we can label it. And now, after interphase, here's my chromosome, my centromere, and the chromosome has copied itself. And it's held together by that centromere. So here's the centromere. And I have 2 sister chromatids. So after interphase, there's still 8 chromosomes, but now there's 16 chromatids because they've copied themselves. Because when this cell divides, they each get 8 chromosomes. Which means there's only 8 chromatids. So mitosis is easy. I have a cell, it copies all of its information. That way, when it divides, each of those cells gets a copy of the information. So I start with 1 cell and then I end with 2 cells that are exactly the same. is a little bit more tricky. I start with a cell. It's got 8 chromosomes. Eight chromatids. After interphase, 8 chromosomes. You might have to copy and paste some more 8s. It's got 16 chromatids. Then that cell divides, 8 chromosomes. And then those 2 cells divide, and it gets smaller. Those divide in half. So now, in meiosis, I start with 1 cell, but I end with 4 cells that are unique. And we're not going to fill in those other chromosome and chromatid numbers. The big thing is in mitosis, I start with 1 cell and end with 2 cells that are the same. In meiosis, I start with 1 cell and end with 4 cells that are unique.